Hello children, welcome to Vivid Botany. The question is about here the nucleases, the enzymes. Nucleases are group of enzymes which cut the nucleic acid and these nucleases are two types. Let us see what they are. Some are exonucleases and some are endonucleases. Exonucleases are those which cut ends of nucleic acid. They cut ends of nucleic acids. Endonucleases are those which cut at specific site of nucleic acid. So what is this specific site? That is our topic today. The specific site is the restriction site at which the restriction endonuclease cuts. So generally the restriction site is also called as palindrome. What is palindrome? Palindrome is that which is same when you read in opposite direction. For example, I write here Malayalam, Malayalam. I write the same in opposite direction, Malayam. So when you check it, it is same. You see M, A, L, A, Y, A, L, A and M. So these are called as palindromes. You can write one more, right? I write here, that is race car, RACECAR. It's also same, you check it, R, A, C, E, C, A, R. So these are palindromes at which the restriction endonuclease will make cuttings, right? Let us see the palindrome or the restriction site of EcoR1. EcoR1 is a restriction endonuclease. It is extracted from Escherichia coli, the bacteria. E stands for genus Escherichia, CO stands for the species coli, and R stands for the strain, that is variety, and 1 is the order. The Roman numerical 1 is order. So EcoR1 restriction site is GAATTC. This is from 5 to 3 direction that I am writing. Now let's write the same on other strand, GAATTC. See, I am writing this on the DNA of two strands. One polarity 5 to 3, the other is 3 to 5. Now you check whether it is palindrome or not. G, A, T, C. So it is same when you read in opposite direction. So how do we know that a particular segment of DNA is palindrome or not? The only simple thing is that it should be same when you read it in opposite direction. Let's make one palindrome. I write one palindrome here. Don't take it that it is really present in DNA, but I will make you understood how can we write, how can we make a palindrome. See, for example, I take it here, T, A, C, G, C, T, A. This is one palindrome. Now let's take the palindrome on other strand, that is T, C, G, T, A, C, G, A. It is 5 to 3. This is 3 to 5. Now let's check in whether it is same or not. You see here T, T, C, C, G, G, then T, T, A, A, C, C, G, G, A, A. This is one palindrome. Like that we can construct any number of palindromes and you can say that whether it is palindrome or not. Because sometimes questions are asked that find out the palindrome among the following. So you should be alert and check it properly whether it is really palindrome or not. Let's write one more palindrome here. Let's take it as C, C, this is G, G, C, T, A. This is 5 to 3, let us suppose. And let us write 3 to 5 also. 3 to 5, that is G, C, A, T, G, C. This is 3 to 5 direction. And check it whether it is same palindrome or not. C, C, G, G, T, T, A, C, C, G, G. So these are the palindromes at which the restriction endonuclease will cut. See, what is restriction? That is also a point of discussion. Restriction means actually the bacteria will produce these restriction endonuclease when they are attacked by viruses. So actually the virus will inject its DNA inside the bacteria, and at that time the bacteria will synthesize, will produce restriction endonuclease to break the viral DNA. So the restriction endonuclease produced by bacteria is aimed at restricting the growth of the virus, restricting the growth of the proliferation, the pathogenicity of the virus. So it is called restriction endonuclease. So where do restriction endonuclease will cut in the palindrome? So these enzymes, that is restriction endonucleases, will cut at ester bonds. They cut ester bonds of two strands. For example, let us take the eco R1. So eco R1 cuts in between G and A from 5 to 3. It makes the cutting of ester bond in between G and A from 5 to 3. So here I take the same diagram, it cuts here and it cuts here. Both the sides it will cut the ester bonds. After cutting, the cut fragments are made like this. Like this they are made. So these fragments are formed called as sticky ends. Let us see here. This is one fragment that is broken and this is the other fragment that is broken. So here if you see fifth end G, C, T, T, A, and here you see A, T, T, C, G. So this is 5 to 3. So this also the 5 to 3 and this is 5 to 3. So these ends are again readily unite. When you keep them together, readily they unite with each other, so that they are called as sticky ends. They are called as sticky ends or they are called as cohesive ends also. They are called sticky ends or they are called as cohesive ends. So with the help of the sticky ends, we can make up a recombinant DNA or a hybrid DNA or a chimeric DNA with the help of DNA ligase. The only thing is that principle you should remember is that when you are going to combine the DNAs of two different organisms, we have to cut the two different organisms' DNA with same restriction into a nucleus to form the same type of cohesive ends. If you cut them with the different restriction into nucleus, what happens? Cohesive ends are not formed. Once cohesive ends are not formed, then they readily cannot unite with each other. That is the case here. Then can you say that here how many hydrogen bonds are broken when a palindrome with cut with eco R1? You see here 2, 2, 2, 2. So total 8 H bonds and 2 ester bonds are broken here. 8 hydrogen bonds and 2 ester bonds are broken when we make use of the eco R1 enzyme. So this is the question I was trying to explain you. Okazaki fragments are those. Let us see the question now. A specific recognition sequence identified by endo nucleases to make cuts at specific positions within the DNA is. It's not degenerate primer. Okazaki sequence are those which are the broken fragments that are formed in lagging strand during DNA replication, and palindromic nucleotide sequences, this is very much correct, option 3. Poly A tail, the adenylate residues, the 200 to 300 attached at third end of hnRNA to form messenger RNA. In the processing of mRNA, they are used. So the answer is here 3. I think you have understood very well my explanation, and my explanation is very much useful to you, very much helpful to you, I am thinking. So let us discuss all the questions one after the other. Please subscribe my channel, that is Vivid Botany. I will be giving you all the questions and respective answers. We don't discuss just the answer, but I will give you an elaborate explanation about the question, and we will see different kinds of questions that can be framed based on the same concept, so that you can have a thorough knowledge and better understanding of the question, and you can solve any type of question that is framed on the same concept. So thank you very much.